Connaissance.
Recherche.
Sciences.
Des sciences mathématiques.
Théorie.
Physique.
Philosophie.
Conférence. Archéologie.
Environnement. Nature.
Thank you, Philippe. Let me first thank the organizers, including the two Philippe and Frédéric, for inviting me to share with you some of the data we have accumulated for the past years about a vaccine strategy against AIDS. I'm saying AIDS, in fact, and not HIV. Knowing the fact that, to me, there are two kinds of vaccines, the ones which are directed against pathogens, and you have some of the examples over there, and the others directed against pathogenicities. As you're all aware, in fact, most of the world is trying to find vaccines against pathogens, and we are probably one of the only teams to actually have some data which indicate that the vaccines against pathogenicity could indeed happen. AIDS is an enigma. It's still an enigma because most of the cells which are dying during the chronic phase of the disease are non-infected cells. And so I started a whole series of experiments with, in fact, two questions and one hypothesis. The first one is, how are these deaths of non-infected cells relate as bystander effect to the viral burden? And the second question was, if the cells are dying, could they be killed? And then came the hypothesis, which I think was a crazy hypothesis at the beginning. Nobody was in my lab wanted to follow me on that hypothesis. And I thought that if the cells were killed, they could not be killed by CTL. And there is another effector cells which could kill, or at least could kill these CD4 T cells non-infected, and that could be the NK cells. As a subsidiary question came the idea that if these cells were able to kill non-infected cells, how it could happen that they don't kill the infected cells? And you will see that we have some results on this matter. So we all started with the idea, and as you're probably aware, but NK cells can kill through a series of NCR receptors, namely NKp30, 44, and 36, and they kill the target cells through the expression of ligands. So having enhanced several probes to detect the various NKp30, 44, and 46 ligand, we surprisingly found, in fact, that a rather high level or percentage of CD4 T cells could indeed express one of those three ligands, namely the NKp44L ligand. And you can see that up to 44% of the cells, depending on the patients, could in fact express this ligand. Two things about this ligand. This ligand is expressed in relationship or in correlationship with the CD4 cell depletion, and you can see that at low level of CD4 T cells there is no expression of ligand, whereas at the low level of CD4 T cells you have this high expression of NKp44L ligand. First point, correlation with CD4 cell depletion. Second point, the NKp44L expression is related to viral load. As you can see here, there is a highly significant correlation with the viral load or viral burden, and you can see that, in fact, up to 40% of cells could be expressed if you have a high viral load between 500 and 1,000 copy by mL. So the first step of our experiment was then to see that NKp44L could be expressed on CD4 T cells. Now, that doesn't say that this molecule is functional. And in the other experiment, which is reported here, you can see that if you make a nice separation of cells which express or not the 44L, you can see that cells which express the 44L ligand are nicely killed by NK cell, where you have no cell killing in blue of cells which don't express the ligand. We went in and then asked the question, how could that be related to the virus itself? This is an experiment when we took normal CD4 T cells, your or mine, and then incubated with inactivated virus. And as you can see here, just by incubation of those CD4 T cells with inactive virus, you have a nice expression of this NKp44L ligand, and then if you dilute the virus, even for minute amount of the virus, you can still have this expression. Next experiment then was to try to detect which type of proteins could in fact be involved in this phenomenon, and we test several of those structures or other type of proteins, and you can see that only two of them, GP160 here and GP41 here. I remember that GP160 is composed of 170 plus 41, and you can see that in fact it's a GP41 which itself is involved in this phenomenon. So it was then easy to try to detect which type of peptide was in fact sufficient to make this phenomenon happen. And in fact we detected a very small motif of only six amino acids in between HR1 and HR2, which are here, just by incubating this peptide with CD4 T cell was sufficient to induce the expression of this NKP44L ligand. Not only it induces expression of this ligand, but it renders those cells sensitive to NK killing. And again, if you incubate CD4 T cell, normal CD4 T cells, with these 6 amino acid peptide from the HIV GP41 involved, you have not only the expression, but the killing of CD4 T cells by the autologous NK cells. Interestingly enough, this motif, which I'll call 3S because there is 3 cysteine residues, is highly conserved, and there are only 3 motifs which have been described in all the series of HIV viruses, and all the 3 motifs, in fact, can do exactly the same type of phenomenon, indicating that it is a very general phenomenon for this type of virus. Interestingly also, and this is a rabbit antiserum, if you have induced a rabbit antiserum, you can totally block this phenomenon, which in fact could be easily envisaged. So antibodies against 3S peptides, as you can see here in diluting amounts, could block this in a phenomenon. So let's resume at that point. A ligand is expressed under just the incubation of a HIV41 peptide together with CD4 T cells, and this phenomenon could be blocked by antibodies. When in fact is this peptide dismarked? Here is an experiment. I go quickly on that. This is 2 viruses which have been incubated, either CCR5 or CCR4 tropic with CD4 T cells, and these are viruses which are deficient in either in the CD4 binding or in the fusion binding site. And you can see here that you still, in those experiments where the virus has fusion deficiency, you still find the phenomenon where it is lost when the virus is mutated for CD4 cell binding, indicating that in fact it's the time when the peptide is demasked is between the CD4 binding and the fusion point, which in fact could be easily envisaged. Now, to say that the 3S peptides could in fact induce the expression of a ligand doesn't tell you how. And this is I'll go quite quickly on that because this is a whole signaling pathway that we have seen just by incubating the 3S molecules together with CD4 T cell. We found, interestingly enough, that in fact this peptide has a receptor which is the GC1Q receptor. By triggering this receptor, it induces a series of signalings which in fact induce the expression of H2O2 without the H2 within the cells. And the ligand is in fact inside the CD4 T cells. So what the signaling is only due is just to make it express at the cell surface. Interestingly, this GC1Q, it's interesting since at the same time, another team of my lab had discovered that with the whole genome scanning, we found that 2 genes seems to be involved in the evolution toward AIDS. One of them was HLA-C, which fits with NK killing, and the other is the complement series, which also fit with that series of data. To go very fast on the question, which is why the infected cells are not then killed if the NK was able to kill NKP44, a ligand expressed of CD4 T cell. When, as you can see here, there is a total exclusion of the expression of the ligand in infected cells. The ligand is only expressed in non-infected cells and not expressed in infected cells, indicating that in fact there is an escape mechanism within the reservoir we went on and in all the series of proteins which in fact could be involved in this phenomenon of escape, you can see what one of them, which is NEF, in fact is sufficient to retain the ligand inside the cells and interfere with signaling pathway, which I showed you previously. This ligand, this phenomenon of NEF escape is interesting because it's totally different from the two other pathways which are that NEF is doing for CD4 and for MHC Cas1. As you can see here, there are one mutant of NEF, in fact, can stop this phenomenon so that, in fact, NKp44L could be expressed at the surface, which is not represented there, as if you block with an anti-NKp44L ligand this phenomenon, you can reduce it to the normal level, indicating that the whole effect of NK killing on CD4 T cell is in fact due to this NEF effect. So to that point, we can conclude that the triase peptide is inducing NKp44L ligand on CD4 T cell. This involves a signaling pathway. It's only expressed on uninfected cells, and there is an escape mechanism on infected cells. Now let's come back to the antibody and also to the project we have at the beginning, which was to set up vaccines, in fact, against this phenomenon. I told you that, in fact, antibodies against 3S, at least rabbit antibodies, were able in vitro to completely block the phenomenon. So I'll show you there two series of experiments, which are ex vivo experiments, where we tested the expression and titers of anti-3S antibodies in human sera. And as you can see, this is interesting because it's a multivariate studies between two series of 40 patients. The first one with a slope of CD4 T cells, which were no slope, I would say, no CD4 cell depletion, and another 40 patients, which is a decrease of CD4 T cells the way it naturally occurred during infected patients. And as you can see, which I think is very interesting, the only predictable factor which makes us to think that the cells will drop down is the antibodies against 3F and not the viral load. So in fact, the prediction is more important, again, the prediction is more important by detecting the antibodies than by measuring the viral load, which I think is quite an interesting point on that. Now, this is a second series of experiments which he did, and showing also that, in fact, on a 3-year survey of patients, you can see that all the patients which in fact have a high titer of CD4 T cells antibodies have no decrease of CD4 T cells, where those which are a decrease of their antibodies have a naturally occurring CD4 cell depletion of CD4 cells per cubic per month. So the summary on that is NKp44L is specifically expressed on non-infected CD4 T cells from HIV patients. It is induced by a specific peptide from the GP41 from HIV-1. Anti-3S antibodies are predictive of the evolution of the disease. And we come to rather maybe two simple scheme, as I can show you. HIV-1, in fact, could at the same time induce the level of antibodies and the expression of the ligand. CD4 T cell is the result of both. When you have a decrease of anti-3S, you have an increase of NKp44L, and then came these effector cells, which are the NK cells. Well, all that is fine. It can fit with the hypothesis, but it doesn't tell you that it's true. So we had to go in experiments. Let's switch now from non-primate macaque models. And first, we have to do this proof of concept that all which I was saying you could at least be, well, at least true or partially true. And we set up a macaque model of HIV infection. I remember you that HIV is in fact have the envelope of the HIV-1. In this model, which I think is remarkable, is that you have exactly the same type of curve that you have in human, which is a rapid drop of CD4 T cell. It goes up to the same level, and then within months it goes down as an evolution of the disease. Interestingly enough, as a mirror image of, in blue, the curve of CD4 T cells, you have this increase of these NKp44 ligands. And this is very clear. If you report now on the right panel, you can see again that you have this increase of NKp44L, but this is, or it seems to be, resulting of the deep drop of antibodies against 3S. So started on that, we tried then to do vaccines in coupling the peptide to KLH and see what happened in the animals. So this is that you have, I think it's 2 years experiment, something like that, and you can induce the level of antibodies with 3 shots of KLH coupled to peptides up to 1 log above the natural antibodies which can occur in these animals. Now, there is absolutely no significant effect on viral load, as it was expected. But there is two important effects on the expression of CD4 T cells and on the drop of CD4. First, as you can see, there is a lack of expression of CD4 T cells. These vaccines totally block the expression of CD4 T cells, where in blue here you have the increase of CD4 T cells in animals which have been infected. Now, as a result, you can see that the level of CD4 T cells is there is a drop in the control animal of these CD4 T cells, where in the animals which have been vaccinated, there is no drop, no depletion of CD4 T cells, which remains exactly at the level they have been before infection and before immunization. And you can see here also that, in fact, with the vaccines, there is no NK lysis, at least no different from animals which have not been infected, whereas you have an increase of this NK killing, as I think I showed you previously. In this right panel, we measure the apoptosis either in peripheral blood or in lymph nodes, and you can see that there is no apoptosis which is detectable in the periphery, at least no different from the control, whereas you can see an apoptosis as a function of time, and a drastic difference of apoptosis in lymph nodes. In other words, vaccines, in fact, could have an effect on CD4 depletion, could block NK lysis, and could block cell apoptosis in the animals. Which is interesting, in fact, is all the animals which are dying, all the cells which are in apoptosis expresses NKp44Ls. You have a difference between cells which are annexin positive and cells which are annexin negative. But that doesn't say that all the cells which express NKp44L are dying, which is expressed here, approximately 1/3. So again, all the cells which are dying express the ligand. All the cells which express the ligand are not dying, indicating that it might be two phenomena, one hit by the 3S and a second hit by NK cells. Interestingly, for me, well, I think you can share that, is the fact that, in fact, not only you have an effect on NK killing, on apoptosis, on CD4 depletion, but you have an effect on activation. And you can see here that you totally reduce the activation of CD4 T cells and greatly reduce the activation of CD8 cells and on CD3 T cells, indicating that, in fact, contrary to what everyone is thinking, we are postulating that activation is a consequence of death and not the contrary. It's not because cells are activated that they are dying, but because they are dying that you have a cell activation. Nobody has ever proven that, but I think these data could indicate or could go in that direction. And it is shown also here. If you look to two inflammatory products like CRP or TNF, you can see that there is a significant reduction of those products as detected in the sera, indicating that, in fact, these vaccines, again, only on 6 amino acids, is sufficiently able to induce a series of phenomena, not only apoptosis, but also activation and inflammation products. Now, this is another series of experiments now, which is a therapeutic, using the vaccine as a therapeutic vaccines. And for me, it's probably one of the most interesting experiments. These animals have been infected 3 months before, and then they are immunized by the KLH-coupled peptide. And you can see that the ligand is expressed around 35-36% of CD4 T cells. Just by immunization, you increase the level of, as you know, you stimulate the viral replication. So you have an increase of expression of NKp44L in the control. But at the same time, in animals which have been vaccinated, you totally decrease the level of NKp44L expression. And then, which I think is interesting here, is that you totally switch the repertoire of NK cells. Those cells are not anymore able to kill CD4 T cells of these animals, where you can see that there is still a nice killing of NKp44L CD4 T cells in those animals. Not only that, but you can see here that in those experiments, there is a stability of CD4 T cells, and in fact, that's correlated to the stability of T cell effector memory central memory T cells, and that the central memory T cells are both related to the expression of NKp44 ligand and NK lysis. Now, we showed also that there is, again, in those animals which have been immunized therapeutically, a nice correlation between the effect of the vaccination on CD4 cell apoptosis and expression of the ligand, as shown here in the lymph nodes and in the rectum, indicating that the phenomena which you can detect in the blood also take place in the tissues. I go on that, which indicates that in fact you block the FAS and Caspase killing. And then again, you find this phenomenon is that just by this therapeutic immunization, you reduce significantly the level of activation on CD4 T cells and also in the tissues. So let me conclude on that. It's maybe too simple, but all the ideas with all the data fits with the idea that the TS peptides is in fact greatly responsible for CD4 cell depletion, and that it is blocked by antibodies against 3S, and that may be the resulting effect of activity of NK cells, CD4 T cell depletion and CD4 cell apoptosis inducing several markers of activation of inflammation. Now, what does that say in summary? I think we can think to three series of conclusions. First, that if we relate to the data we have on NKp44L expression and NK and NKp44L correlation, interaction, you would agree with me that at least in vitro and as I showed you in vivo, we think that the CD4 depletion of non-infected cells is mostly due to this phenomenon, and that may explain the bystander effect which is observed. Now, interestingly, there is a preserved CD4 cell reservoir, and this is one of the genius of this virus, of the infected cells by an escape mechanism. From the immunological point of view, I must say I've been quite interested, maybe more than that, but all of the data, that in fact, and I think this is one of the very few experiments which demonstrate that with the specific vaccines, you can control innate immunity. And not only you can control innate immunity, but you can induce an immune memory which can control innate immunity. So this, I think, to the future of vaccines is quite an interesting point. In relationship between now infection and this bystander effect of death of uninfected cells, it's a nice balance, in fact, for viral chronicity, and we could discuss what would be the resulting effect of the balance between reservoir and infection. But that's also demonstrated that maybe the virus has been selected on two ideas, one for infectivity and the other for a deleterious effect on CD4 T cells. So maybe I push it too much, but the idea of a virus which could have two types of selection, one to kill and one to remain in the reservoir, is something which I think is interesting. Now, by the way, we demonstrate that in fact there is a pathogenic epitope which on the virus. If we go now in immunotherapeutic interventions, I've seen you that in fact, at least in these two series of experiments as a preventive or therapeutic vaccines, there is an effect on the evolution of a series of markers of AIDS, and that these experiments are going into clinics next year, in fact. We think that it could be useful, not competing with the other one, which deal with infectivity, but this may be one of the very first example of a vaccines, in fact, which could be directed against the disease and could be used to delay drug use. And this is, as you all know, very important, especially in the south, where the disease is progressing, to prevent inflammatory process in patients. And also we know now that there is approximately 15-20% of individuals which, in fact, although they are good viral responders, are in fact immunological bad responders. And we have data indicating that in those conditions, in fact, these individuals which are immunological bad responders do express these ligands and have a lack of antibodies. So we can hope that the vaccines could be helpful in those individuals. Now, I'll end by people who work with that, but I'll say that in my lab, only one of my collaborators was able to follow me at the very beginning of the hypothesis. And practically all the work has been conducted with him. All the experiments in the macaques have been done with Roger Legrand and Nathalie Bosquet and some other parts of the world with Olivier Schwartz. Thank you.